fast-growing fingernails, whatever it might be. If we can use it to identify a characteristic of an organism, then we call it a gene. Okay, a gene is like a cluster of bases that we know does something or identifies a characteristic in the organism. That's a gene. It's a cluster of base pairs. The genes are found in the chromosomes in the nucleus. Human beings have 23 of them. Different animals have different amounts, but human beings have 23 chromosomes. And during the reproductive process, at least in in meiosis, in mitosis it's slightly different. But let's look at meiosis here, which is the standard reproductive process that human beings undergo. Anyway, so in the reproductive process, the DNA strands of each parent, the DNA splits. Remember, there are two strands. It's a double-stranded DNA. That's a standard DNA. It splits into two different strands, and each strand can combine. And when the parents get together, a strand of one can combine with a strand of one parent can combine with a strand of the other parent. So we have some kind of new DNA that comes from the combination of DNA from each parent. So in this way, the offspring is different from each parent. Now, this is a very, very overgeneralized, simplified version of reproduction using DNA. The main point is that when you have two parents, there's some DNA that comes from each parent. The other biological relevance of DNA is that three base pairs, just a group of three, they actually are a code for produce this amino acid. Again, it's the blueprint, it's the plan. So it says produce this amino acid. Amino acids build proteins. So if you have a bunch of base pairs, you're building a bunch of amino acids together and you're going to make a protein. Proteins are the workhorses. They actually do the work. They do the work. They provide building materials. They gather building materials. So we have two things: the DNA, which decides what to do, and the creation of proteins, which actually does it. So that is like the entire organism. The DNA, which has the plan, the protein, which actually does the work. Again, overgeneralization of everything, but this is enough for us. The group of three base pairs provides a code for the amino acid, and therefore several groups of them will produce the code for the protein. We know the code of these three base pairs. We know, let's say, AGC produces tyrosine or whatever. I'm just guessing at these things. We know the codes, which three base pairs produce which amino acids, such that it will produce a certain protein. We know that. However, there are many segments of DNA there where we just don't know what it's doing, which is normal enough. I mean, do we understand every single thing about a human being? So certainly parts of DNA, we don't know what exactly they're doing. So some of these segments they're considered, they seem to do absolutely nothing. And they're called junk DNA. Now, it's not a simple thing in the biological world to say, oh yeah, that's this thing that the creature has and keeps as it reproduces is useless. That's not often found in the biological world. So to say that a large part of our DNA is useless, people are not so sure about it. Maybe there is some use to this, what's called junk DNA. We don't know the use, but that doesn't mean it doesn't have a use. And they're debating whether there is a use, or maybe it was just some accidental thing that got into our DNA. Here's a very important idea: how to replicate DNA. Now again, human beings, when their cells divide with mitosis, it just the DNA splits, and they splits. When it comes to producing offspring, the two parents they split. Two parents get together, they rejoin in a different to form an offspring with different characteristics. Okay, but now let's talk about how it's done in the lab. How to replicate DNA in a lab, because that's what in the forensics lab is done. We take a tiny amount of DNA, we replicate it, so we have plenty of it. So what we use is what's called the polymerase chain reaction. Polymerase is an enzyme. The ACE implies that something is an enzyme. If you hear something called a PET-ACE, well, you know that kind of thing is an enzyme that works on PET. PET is polyethylene tetraphthalate, It's what makes a lot of our plastic that we use and we throw out. So if we can develop some enzyme that'll eat up all that plastic, we'll have many fewer problems with our garbage. And it's true, they actually did make PETase. That's called PETase. It's an enzyme that works on PET, polyethylene tetraphthalate. Anyway, so this polymerase is an enzyme, and what it does, it doesn't break things apart, it actually enables DNA to replicate. It does this during the natural cell replication, but we can use it in the lab to take DNA that we find and replicate that, or even just parts of it. What it does in general, we'll see how it works in more detail soon, but if you've got DNA, you put it in a solution with lots of the free base, all kinds of A, the T, the G, the C, all over the solution. A bunch of phosphates, a bunch of deoxyribose, fill the whole thing with all that stuff in the solution, and you put this polymerase in there. You will enable that DNA to grab all the bases, phosphates, and replicate itself and form more and more and more. So let's see how this is done. Here we go. Let's start with double-stranded DNA. Here's AGTCC. That's our strand. The complementary strand always has one for double-stranded DNA is TCAGG. In case it wasn't mentioned, the way this thing arranges itself in space is in a double helix. So we have the double helical structure of DNA that you might have heard about. We're not going to deal with the double helix. So just keep in mind that we have one strand over here and a complementary strand over there, and they stuck to each other. That's natural DNA. That's what your people have. That's what people are going to deposit at crime scenes. Double-stranded DNA. Now, if you heat it up enough, not to the boiling point, just below it, the boiling point is 100 degrees Celsius. This is 94 degrees Celsius. Heat it up to this temperature. We certainly can do that. And we will actually split the strands from each other. Now, each strand will stick to each other very well. This will not break. But what connects the two strands, one strand to another strand, that will be broken apart. So now you have two different single-stranded DNA. Remember, we put this, this is all in a solution where we got all kinds of stuff that's needed. All kinds of raw material. You got the A's, the T's, the G's, the C's all over it. You got the phosphates, you got the deoxyribose, you got everything you need floating around. So we got all this stuff floating around, A's, T's, all this stuff. When you add the DNA polymerase, this is the enzyme at 72 degrees Celsius, the polymerase will put the T's next to the A's and the G's next to the C's. It will arrange all the raw materials that are floating around there into the right place. T next to the A, G's. It will arrange it. Now remember, we had two strands. We started with one. We got a double strand. It'll arrange these around the strand to the left. These will arrange the strand to the right. And of course, it doesn't just do that. It arranges the phosphates and the deoxyribose. So it'll actually form complementary strands for each. So what's left after the DNA polymerase is done is we have two strands. We started off with a single double strand. Now we have two double-stranded DNA. We have actually copied the original DNA. These are exact copies of the original. The polymerase doesn't disappear. After it arranges everything, it moves away and gets ready for the next one. So only is a little bit of this DNA polymerase. It acts as a catalyst, so it doesn't really get destroyed. And we've copied the DNA into two identical strands. This part gets done again. These two copies, we have them, heat them up. Each one gets split. It's still all this solution of all the raw materials. DNA polymerase, we'll stick them back together.